Hey everybody! Today we're going to be talking about a wet mount technique that we're going to be using in our class. And when it comes to wet mounts, these are generally just little microscope techniques that we can use if we have a sort of sample from a body of water. So whether that's like the ocean or it might be like a pond or something like that, if we kind of want to know if there's any type of microbes in that sample, we can do what we just call a wet mount and then just look at it under the microscope in order to see if we can find anything that's kind of moving around. So the materials that we need in order to perform a wet mount, it's very limited. You're going to need a slide. You're going to need a little cover slip, just like this tiny little either plastic or glass piece. You're going to need a sample, and then you're going to need a pipette in order to collect a sample with. So just those four basic items there. Of course, you're going to need the microscope too to look at it, but these are the basic things to do our technique here. So in terms of the overall procedure that we use, it's also really straightforward. Got your microscope slide, stir up your sample a little bit to kind of get a lot of those small little things kind of moving around. Collect some of it inside of the pipette, and then just kind of squeeze out a little bit. Is generally going to give you a better result. And then just one drop right on top of your slide. After you've done that, we're going to take our little cover slip and we're going to sort of hold it at a 45-degree angle over the water, and then we're going to sort of squeeze it down. And basically what that does is that any sort of air that is located inside of our water is then being pushed out. So at the end, again, we just kind of get something like that. We got our little cover slip on there with our water under it. And that's basically it for the procedure. Now you have made a wet mount. And then what we can do is we can go ahead and we can look at it under the microscope. And when we're looking at it under the microscope, there are going to be lots of different things that we're going to be seeing again, because this is going to be a very mixed sample. So a lot of times what we might see is like small little protists are going to be in there, or even certain tiny animals like little water fleas and things like that are probably going to be inside of your sample. But we're also going to see a lot of debris in there, so there's going to be a lot of plant material that's going to be stuck a lot of times when we do these pond water samples. Or there might be cracks within either the cover slip or the glass that you might see. So normally, in order for us to sort of determine whether or not we're looking at like a living thing, a lot of times we try to look for movement, or what we call true motility. So kind of like in the video that I have here, what we're following is most likely just a little ciliate. And you can kind of see how it's like bouncing around and it's sort of moving around with purpose. It's not just kind of flowing from one side of the lens to the other. It's actually sort of moving around and bouncing all over the place. And this purposeful sporadic movement is what we call true motility. This generally indicates again that this is a living organism because it is actively moving to different parts of our slide, either to get into safety or to find food is kind of the common things that we're looking for there. Now, we can't confuse, again, this movement, though, with what we call Brownian movement. And a Brownian movement is kind of this flowing river that we see, or this sort of just kind of a flow of molecules as they move through the water. So kind of like in this video right here, what we're looking at are little air bubbles, basically. And you can kind of even tell that as I'm moving the lens in order to keep up with, again, this kind of flow, it's almost like I'm pushing those little bubbles along. And again, this just kind of shows us Brownian movement. It's just a sort of natural flowing movement of different types of items as they just flow through water, basically. Now, the reason why we actually like to run a lot of these wet mounts and things like that is that it's a good addition to our microscope practice that we do in class. And the big thing here is that a lot of the stuff that we've done so far has just been looking at samples where we know that there's going to be something on the slide. But when we do an actual wet mount, again, we're not really sure what we're going to find. There might be a lot of stuff in it, there may be nothing in it. So it's a good practice, again, to kind of look for these items that are not going to be colorized, so they're not going to have as much contrast. And it really just kind of helps us really nail down and figure out if we have that good microscope technique that we need for our class. But other than that, I don't think I have anything else to cover on how to do a wet mount.